Hi, welcome to the Sybil Cluster Search Page Screencast. In this screencast, we'll be covering the features of the cluster search page. It is helpful if you've already watched the protein search page screencast, as these two pages are quite similar, and there are elements that I will not be covering again here. Let's get started. We'll begin at the strep-pneumo Sybil homepage at strep-pneumo-sybil.igs.umaryland.edu. From here, we'll click on the button labeled Protein Cluster Search. This brings us to the search page. We'll need to click on the tab in the upper left-hand corner labeled Search for Clusters. Like the protein search page, the cluster search page is divided into two sections: the search form on the left and the results area on the right. To start, we'll focus on the search form on the left. The first thing that must be decided when searching for gene clusters is which cluster set to search. In this case, you can choose either ortholog or paralog clusters. The clusters currently in strep-pneumo are derived using bidirectional best blast hits. Next is the gene product name filter. This filter can require just one member of the cluster to have a matching gene product, or some percentage of the members. The protein length filter allows filtering by minimum length, maximum length, or both. Filtering can either be required by all members of the cluster or just by one member. The next two sections allow filtering of clusters by number of proteins and organisms respectively. Setting the representative annotation source determines which organism will provide the displayed gene product name for each cluster. If the selected genome does not have a member in the cluster, a name is chosen at random. The last section allows filtering based on organism membership. There are three tables: must include, no preference, and must exclude. Organisms can be dragged into the different tables to filter clusters based on whether or not they contain a member gene from a particular organism. There are shortcut buttons at the bottom of each table to quickly move all the members of a table to an adjacent table. Multiple organisms can be selected using the Shift, Control, or Command keys. Let's try it out. I'm going to search for ortholog clusters that contain the word protease in the gene product name. I'll only require that one member of the cluster contain the word protease. I'm going to choose Tiger4 as my representative annotation source. And I'm going to ask for only clusters that include Tiger4. And do not include Hungary 19A-6. Now I'll click Search. So I see in my results area that I have two clusters that contain the term protease and are present in Tiger4 but not in Hungary 19A-6. Like the protein search page, I can collapse the search form by clicking the double left arrow at the top right-hand corner of the form panel. The columns in the output area are number, which represents the original ordering of the results, name, which is the unique identifier for this cluster and is a link to the cluster's report page, representative annotation, which is the gene product name from the representative annotation source, total number of organisms, which represents the total number of organisms in the cluster, total number of proteins, which is the total number of proteins in the cluster, average seq length, which is the average protein sequence length in amino acids. The next two columns, leading and trailing gaps, indicate whether or not the multiple sequence alignment of the cluster members has gaps at the beginning or end. No indicates no gaps, and yes indicates gaps. The min seq length over max seq length ratio gives an indication of how divergent the lengths of the cluster members are. Percent identity and percent coverage columns indicate the average values calculated for all the pairwise alignments in the cluster. This table has the same controls as the protein search table with regard to sorting, reordering columns, etc. The only difference is that there are columns that are hidden by default. To show these columns, hover over the column header and then click the dark down arrow at the right-hand side of the header. This reveals a drop-down. Go to the columns entry and you will see the filtered number of organisms, filtered number of proteins, length variance, and length standard deviation are hidden. These columns can be shown by checking their respective boxes. The filtered number of organisms and filtered number of proteins columns are counts of organisms and proteins found in this cluster from the list of organisms specified in the Must Include table. Currently, the number of organisms will be the same as the number of organisms listed in the Must Include table. The length variance and length standard deviation offer another measure of protein sequence length variability within the cluster. The bottom of the output area houses the pagination controls on the far left, the record count and currently displayed records on the bottom right, and 3 download buttons. The first download button is the Tab Delimited button, and it downloads a tab delimited version of the list currently being displayed. The only difference is that all columns will be visible. The detailed text file downloads a list containing both the clusters and the protein members for each cluster. The Organism Profile download button will download a file with organism names across the top, cluster names along the left side, and a 0 or 1 to denote whether or not this cluster contains a protein from this particular organism.